Hello, I'm Professor Matthew Schmidt, and welcome to Genetics. We're continuing on our exploration of recombinant DNA and its applications, and in this session we'd like to look at the construction and usage of genetic libraries. So, what a library actually is, I know you know what the regular one is, the big building, but it's a collection of clones, and they don't have to be, but we're going to say that those clones are individual plasmids, each of which carries a single portion of the genome of a particular organism. So in other words, the net sum of all of these plasmids together would represent all of the information in the genome of the organism. You can see the analogy of a library if you say that each of the books in a library represents sort of the same idea as one of the clones here, and that the whole bunch of books in the library represents all of the knowledge that exists, something like that anyway. So you can construct lots of types of libraries. The most inclusive one, and the kind we'll discuss first, is called a genomic library, meaning that somewhere in those books in the library, those clones, you will find every single gene and other DNA sequence present in the genome. So I think I just said this, but all of the clones taken together contain at least one copy of every DNA sequence in the genome. But you might say, so what? What's the difference between having that and the genome? Well, we're about to find out. You construct the library using the same principles as we used in cloning of specific genes. So we can just remember those tools and things that we used. So how would you do this? And then we can discuss why you might want to do it. The first thing you do is isolate total genomic DNA from an organism. Let's just say it was a human to go along with what we've been doing. Now in this case, you'd want to digest that genomic DNA with a restriction enzyme. It doesn't particularly matter which one in this case, but what you can imagine that would do is create literally thousands, if not more, fragments, depending on the size of the genome, the frequency of a particular restriction enzyme recognition site. In fact, you can use those variables if you want to have more or less fragments. And then, so you've got all these different fragments. Incubate the fragments with appropriate cloning vectors in the presence of ligase. So it's almost as if we're doing what we did last time when we cloned a gene, just on a large scale. So the bottom line is you're just taking all the fragments, potentially hoping that they all get in to a single, each of them gets into a single cloning vector, meaning you might have thousands of plasmids, but each of them took up one of the pieces that we created when we restriction digested the entire genome. And as we saw before, then you would use the transformation process to introduce these many recombinant plasmids into bacterial hosts. So what the result would be is a collection of clones, but really what it would be is a collection of clones inside of bacterial cells representing every single sequence in the genome. So in other words, one cell may have taken up a recombinant plasmid that includes one particular gene. We'll have another cell that's taken up a different one, etc., etc., etc. So here's a great picture, I think, that explains, well, pictorially how to do this. So it doesn't have to be, but we're going to take a person here, human DNA, and you have total genomic DNA. You could get it from any place that it exists, which is every cell in your body pretty much. And we're going to create recombinant DNA molecules. Well, first thing we're going to do is chop it up, right? Using a restriction enzyme. And in this case, you can see that all the blue fragments that are over here represent, there will be a lot more of them, but they represent pieces that were made by chopping up the human genome with the restriction enzyme. Then we're going to take plasmid DNA, which is going to be our cloning vector, and we're going to digest that with the same restriction enzyme, right? So the red fragments over here represent the fact that actually what they represent is that these cloning vectors should have one site for the relevant restriction enzyme, right? So they're really, they're linear pieces there, but what we've really done is opened each of them up so that it's ready to accept one of the blue fragments, in this case, one of the pieces of the human genome. Okay, so now we're ready to look over here. We're doing basically, I hate to keep saying it, but what we did before, which is create recombinant DNA molecules, in other words, plasmids harboring the human sequences, by joining the fragments together with ligase, just like we did. And what we'll end up with is a population of plasmids now sort of randomly containing different pieces of the human genome. You could see here, now what you can see here is that, look, some of the plasmids did not in fact take up a piece. This one took up one piece, this one took up another, etc., etc., etc. Then we could take E. coli. I mean, you don't have to use E. coli, but everybody does since it's readily available and we know everything about it. Transform various cells with these recombinant plasmids. So you'll then have, and do the whole thing, transform, select, so you know which one's got the plasmid, and then you'll end up with a whole collection down here of bacterial cells, each of which harbors a plasmid, and each of those plasmids harbors one portion of the human genome. So I just wrote down here for fun, this is maybe this is where our human insulin gene that we were working with last time came from in the first place. This fragment happened to harbor the human insulin gene, so that ends up in one of our clones. The CFTR gene is the one that, when mutated, causes cystic fibrosis, so maybe that ends up in another place. And I wrote question marks, meaning we don't know. But what we do know is that every single gene and other sequence in the human genome is represented somewhere in that library. So I think that's understandable. What are you going to do about it? Do with it, I should say. Well, before we ask that, I mean, partially we're going to address these things in the applications session. But before we ask that, there are some other types of libraries that you should be aware of and understand the difference. So chromosomal libraries are pretty popular. And if you've already narrowed down a gene's location to a particular chromosome, and you can do that by more traditional mapping techniques often, or there are a variety of ways, what you can do is just get a library of sequences from that chromosome. If you know you're interested in a particular gene, in humans obviously there are 23 different chromosomes. Say you knew that your gene of interest was on chromosome 7. So if you get a library just of all the material in chromosome 7, you don't have to parse through all of the information on the other chromosomes. A very important and interesting type of library, and just a concept in general, is that of a cDNA library. cDNA stands for complementary DNA. So if you're looking for a protein coding gene, which you're not always, but you often are, you can save yourself a lot of time by using cDNAs. So protein coding genes are expressed, and if they're expressed, there must be a messenger RNA that's made, right, to make the protein. Now in humans this would be tissue specific, and you can use that as well. In other words, you could make a cDNA library from the liver, which would have all of the proteins. I should say what it would have is all of the DNA sequences corresponding to proteins that are expressed in the liver. But how you do it is really cool. You get the relevant messenger RNAs and then use the enzyme reverse transcriptase, which naturally occurs in retroviruses, to backwards transcribe and make that RNA into DNA, but it's cDNA. And look at the beauty of this. Not only are you gotten rid of all the extraneous genomic sequences, I say. In other words, we haven't discussed this a lot, but sometimes the term junk DNA is used. I don't like that, but let's just say that the majority of sequences in the human genome don't in fact encode genes that make proteins. They do other things, or maybe they don't, but there's a lot of material that is not relevant if you know that that's what you're looking for. But also another